Hey guys, welcome to the very last lesson in Chapter 7. This lesson is going to focus on almost entirely about how water comes into this process. Last time I talked about how you'll be able to describe how things come into and come out of the cell, but I didn't talk about water very much. And water is extremely important in this whole process. So I'm going to go ahead and spend most of today's lesson talking about how your cells handle water and the problems that might become associated with water. So, diffusion of water is just called osmosis. So, water is a solvent, and that means that water likes to dissolve things. So, if you ever put salt into water and then stir it, what happens is the salt disappears, it becomes dissolved in the water. The same thing happens with sugar and other molecules as well, and proteins. Proteins dissolve in water. Now, what happens in osmosis is that osmosis is the movement of water from one location through another. So here's a quick little example right here. If I have a selective permeable membrane right here, and I have water, an equal amount of water on both sides, but this side here has more solutes, things like sugar and salt, than this side here does. What's going to happen is that the water is going to move through the membrane until both sides have an equal concentration of solutes. So now both of these sides have the same amount of sugar molecules and salt molecules, but they have a different amount of water. So their concentration has equaled themselves out. This is called osmosis, and it's really important for cells. And here's why it matters. Water is just like any other substance. It moves from areas of high concentration to low concentration. The problem is that water is very small as a molecule. It's only one oxygen and two hydrogens. It's a very, very small molecule, which means it's very easy for it to pass through the plasma membrane pretty much without a problem. So if too much water moves into a cell, the cell literally explodes. And I'll actually show you some pictures of cells exploding because they had too much water. The opposite is also true. If you have too little water inside the cell dies because the cell needs to have water to survive. So cells have to balance how much water gets in and gets out. Now, your body does this by using the same kind of channel proteins to try to bring in more water and try to get rid of water when it needs to. But even despite the fact it isn't always able to balance it out, we have three ways that we can describe cells in water. And I'm not going to lie to you guys. These are really confusing. They're going to take some time to understand. And I can tell you that I have to constantly review these three ideas. If I don't, I forget them because they don't always make common sense to me. But once you understand it, they do actually make sense. So, an isotonic solution. An isotonic solution means that the concentration of solutes in the water is the same inside and outside the cell. So, basically what that means is that everything's pretty equal. So you see this, there's the same amount of water coming into the cell as is leaving the cell. There's the same number of solutes, things like sugar and salt, outside of the cell as there is inside of the cell. This is called isotonic. Here's two pictures here of cells that are in isotonic solutions. These are red blood cells right here, and they look normal. They look healthy. These are red blood cells that are in an isotonic solution. These plant cells here, you've probably never seen plant cells before, but these are very healthy looking plant cells. And you'll see some examples of very unhealthy looking plant cells here in a minute. Now, the next type of situation is something we call a hypotonic solution. What that basically means is that there are more solutes inside of the cell than there are outside of the cell. Now, What happens in hypotonic situations is that water rushes into the cell and a whole bunch of water gets pushed in there. Why this is a problem is, as you can see right here, these cells right here are the exact same as these cells right here. Can you see a difference? Oh yeah, it's a big one. These cells right here are big and they're exploding. They look like they're ready to explode. And in fact, some of them have exploded. So in a hypotonic solution, there's too many solutes inside of the cell, and the cell has water rushing into it, and that water literally causes the cell to explode. You can see the same thing here in the plant cell. If you look carefully here, in the plant cell, most of the stuff in the plant cell has been pushed on the outside of the cell now. If you look here, everything's pretty equal. That's because water has rushed into the cell. So being hypotonic is a bad thing. You don't want your cell to explode. The next one is called a hypertonic. Hypertonic is the opposite problem. That means you have too many solutes outside of the cell and not enough inside of the cell. So what happens here is the exact opposite problem, is that water leaves the cell, and what ends up happening is the cell gets basically crushed. If you look right here, these are again the exact same cells that we saw before. We look how they look, they've all been kind of crushed in on themselves. That's because water has left them and they're losing their water. And as a result, they're going to die. Both of these situations, a hypotonic and a hypertonic situation, are very bad. So your body has to regulate hypertonic and hypotonic and hypertonic, otherwise the cell is going to die. So your body wants to have isotonic. Isotonic is perfect. This is what it wants. This is what it wants to maintain. So your body spends a lot of energy making sure that things stay isotonic and they don't become hypotonic or hypertonic. And you can see in the pictures how this is a bad thing. So here's what I want you guys to do now. I want you guys to go online. I want you guys to grab the Virtual Membrane Diffusion Lab from either from the flash drive that I gave you, the folder that was in there, or from Edmodo. And I want you to complete this lab. This lab is a simulation-based lab, meaning everything is done on a computer via the PhET website. This lab will probably take you guys about a lesson and a half to complete. So if it takes you a while, don't feel bad. At the very end of this lab, it wants you to create a table, or sorry, a graph. You have two choices on how you do this. I would prefer you choose the first one. The one I prefer you guys to do is I want you guys to make an Excel graph. I want you guys to make a graph on Excel that shows your guys' data. You can also create one on paper and then take a picture of that and post that picture onto Evernote, but I want you guys to learn how to use Excel. So I'm hoping that you guys be willing to ask me for help on this or talk to your partners as well and find out how to make tables on Excel, because it's a very good skill to have and you'll definitely be useful for you later. When you're finished with this lab, guys, I want you to turn it in onto Evernote, and that's it. Thank you for watching, and good luck.